Assalamu alaikum warahmatullahi ta'ala barakatuh. I'm Selma al-Mahdi, and today we are going to discuss oxidation of fatty acids. Our objectives will be to list, compare and contrast different types of fatty acid oxidation, to state the significance of fatty acid oxidation in energy metabolism, to identify the cellular locations where fatty acid oxidation occurs, to define a specific type of fatty acid oxidation that is called beta oxidation, describe the steps of activation and transport that precede the reactions of beta oxidation, to list the reactions and enzymes of beta oxidation, to calculate how much ATPs are produced from complete oxidation of palmitate, to discuss the regulation of fatty acid, and to discuss some examples of the disorders of fatty acid oxidation. Marathon runners, birds that migrate for miles, sometimes cross continents, grizzly bears that hibernate for months. What do they have in common? They use fatty acid oxidation as a sustained source of energy for these prolonged activities. Many animals, in fact, depend on fat stores during hibernation, migratory periods, etc. Grizzly bears remain in continuous state of dormancy for up to 7 months, during which they do not eat, drink, urinate, or defecate for months at a time. They carry out beta oxidation in their sleep. What is beta oxidation? Beta oxidation is one of 3 types of fatty acid oxidation that also include alpha oxidation and omega oxidation. And the naming refers to the position of the site at which the carbon atom the reaction will start. For example, alpha oxidation starts in the alpha carbon atom of the fatty chain, which is the second carbon from the carboxyl group, and beta oxidation occurs at the third carbon, which is the beta carbon, and of course omega and omega carbon or the last terminal carbon of a fatty acid chain. Where are the locations at the level of the cell? Beta oxidation primarily occurs in the mitochondria. A specific type of beta oxidation also occurs in the peroxisome. Alpha oxidation primarily occurs in the peroxisomes, while omega oxidation takes place in the endoplasmic reticulum. Beta oxidation is the major pathway for fatty acid oxidation for energy production, particularly during fasting or prolonged exercise. And the substrate for beta oxidation is straight chain fatty acids. Whether this straight chain is saturated or unsaturated doesn't matter, it will be a substrate for beta oxidation. The initial site, as we said, is the beta carbon, and 2 carbon atoms will be cleaved per cycle of beta oxidation. And these two carbon atoms will be released as acetyl-CoA. The energy yield in form of ATP of beta oxidation is quite high. The second type of oxidation, alpha oxidation, is for branched chain fatty acids such as phytanic acid. Alpha oxidation helps in handling the branched fatty acids which cannot be metabolized by beta oxidation, because having a methyl carbon at the beta methyl group at the beta carbon blocks beta oxidation. And in alpha oxidation there will be removal of 1 carbon atom as carbon dioxide, and the products will be a shortened fatty acid chain which is called peristaltic acid plus carbon dioxide. The energy yield of oxidation is quite low or none, because it is primarily concerned with detoxification rather than energy production. Omega oxidation is a minor alternative pathway for beta oxidation. The significance of omega oxidation becomes more when beta oxidation itself is impaired. So omega oxidation is in fact a backup pathway for omega oxidation. And in omega oxidation, there is oxidation of medium to short chain fatty acids, whereby the terminal methyl carbon will be oxidized to a carboxyl group. So now the fatty acid will have 2 carboxyl groups. It is initial one and another one that is introduced by omega oxidation. So the product will be a dicarboxylic acid, an acid that has 2 carboxyl groups. Likewise, the energy yield of omega oxidation is quite low, because its primary concern is not energy production. We will discuss in detail beta oxidation of fatty acid because beta oxidation is quantitatively the most important pathway. By definition, beta oxidation is a catabolic process that takes place in the mitochondrial peroxisome involving successive removal of two carbon units from the acyl derivative in the form of acetyl-CoA, which is further oxidized in the citric acid cycle as reducing equivalents will be transporting the electron transport chain to produce ATP. Beta oxidation, in order to carry out the proper reactions of beta oxidation, fatty acid must be activated first, and then they have to cross the inner mitochondrial membrane before proceeding to the proper reactions of beta oxidation. So a fatty acid will have to be activated first, and then it will have to cross the mitochondrial membrane. Inside the mitochondria, the acyl-CoA of the fatty acid residue will undergo 4 reactions of beta oxidation to be completely oxidized to acetyl-CoA. Acetyl-CoA will precede the citric acid cycle for complete oxidation, while the reducing equivalent that has been produced in beta oxidation will proceed to the respiratory chain to be coupled to oxidative phosphorylation and formation of ATP. The activation process, we mean converting a free fatty acid to an acyl-CoA by adding CoA. This step is the only energy requiring step in fatty acid. It is catalyzed by acyl-CoA phosphatase or thiokinase. As you can see here, ATP is converted to AMP. So we have removed 2 high phosphate energy. Instead of having ABB, we are having AMP. And our fatty acid is now an acyl-CoA with a CoA attached to it. And for the crossing, the length of the fatty acid determines its ability to enter the mitochondria. Fatty acids that are 10 carbon in length or less enter the matrix freely. Fatty acids that are 12 to 18 carbon atom in length have to use carnitine transporter. Fatty acid chains that are more than 18 carbon atom in length cannot enter the mitochondrial matrix. They have to be shortened elsewhere before they can gain entry into the mitochondria. Crossing for the chain length that is 12 to 18 carbon atom is a 3-step process. In the first step, there is formation of what we call carnitine derivatives. Carnitine derivatives are our acyl or CoA, in which the CoA will be converted or removed to be replaced by carnitine. And this is catalyzed by carnitine palmitoyl transferase 1, CPT1. This enzyme is very important. We'll come to it later as we discuss regulation. It is present in the outer mitochondrial membrane. The second step of crossing is transporting the acyl carnitine across the inner mitochondrial membrane, and here we employ the enzyme acyl carnitine translocase, which is actually an exchange transporter that will help in getting acyl carnitine inside the mitochondria in exchange for getting carnitine outside the mitochondria. In the third step, there is reformation of acyl-CoA by a carnitine palmitoyl transferase 2 with libration of carnitine. This schematic representation here nicely demonstrates the steps of activation crossing into the mitochondria. Here is our free fatty acid that is activated to acyl-CoA by acyl-CoA synthetase using ATP and CoA. Acyl-CoA by carnitine palmitoyl transferase 1 will be converted from acyl-CoA to acyl-carnitine with the release of CoA. Acyl-carnitine then can cross inner mitochondrial membrane by the acyl-carnitine translocase. We said it is an exchange transported. It will help acyl carnitine get in, while carnitine in exchange will be forced out. This acyl carnitine here will be under the influence of carnitine palmitoyl transferase 2, which will release the free acyl CoA, which is now ready to go for the proper reactions of beta oxidation. And carnitine also will be released to act for the transport and exchange transport for another molecule of acyl CoA. The proper reactions of beta oxidation are 4 reactions. And in beta oxidation, 2 carbon atoms are removed each time from from acyl CoA starting at the carboxyl end. We have 4 successive reactions that go back to back, releasing 2 carbon atoms at a time as acetyl CoA. The chain of the acyl residue is broken between the alpha and the beta, that is to say between the second and the third carbon atoms, and eventually complete oxidation of palmitoyl CoA through beta oxidation will yield 18 acetyl CoAs. This is our palmitoyl CoA, and this is the carboxyl end. This is the alpha carbon and the beta carbon. In between them there will be a cut releasing the 2 carbon atoms as acetyl CoA. That we need, of course, CoA here as an active part of the molecule, and with successive removal of acyl CoA each time 2 molecules will be removed until the whole 16 carbon atoms are broken into 18 acetyl CoAs. The 4 reactions of the beta oxidation cycle include dehydrogenation, hydration, dehydrogenation, and thylated cleavage in that order. The first reaction, the dehydrogenation of acyl CoA, will remove 2 carbon atoms from the beta and alpha carbon atoms. They will be received by FAD to form FADH2, which will proceed to the electron transport chain. And this will create a double bond, and our product here is a delta 2 trans enoyl CoA, and the enzyme is acyl CoA dehydrogenase. The second reaction, is a hydration reaction where a molecule of water is produced by delta 2 enoyl CoA hydratase to saturate the double bond to produce 3-hydroxyacyl CoA, which will be dehydrogenated to 3-ketoacyl CoA using NAD as a recipient of hydrogens that will be removed from the third carbon atom here, from this hydrogen and this hydrogen, to create a double bond by 3-hydroxyacyl CoA dehydrogenase. NADH2 will proceed to the electron transport chain. The fourth reaction is a thylated cleavage of 3-ketoacyl CoA, in which CoA here will be needed for the new acetyl CoA. So the cleavage is here. These 2 carbon atoms with CoA will form acetyl CoA. Our acyl residue will now be 2 carbon atoms less than the initial length. This acetyl CoA can, of course, proceed to the citric acid cycle, whereas the acyl CoA will repeat the reactions of beta oxidation again, starting from the dehydrogenation. Of course, the step of crossing over, the step of activation are no longer needed. We just repeat the 4 reactions of beta oxidation until the whole molecule is oxidized to acetyl CoAs. The overall reaction of complete oxidation palmitate is that we have our 16 carbon atom palmitate. We need ATP for the source of energy. We are going to, of course, require 2 high energy phosphate. We need 8 acetyl CoAs, 1 for the initial reaction, for the newly formed acetyl CoAs. We need NAD and FADs as coenzymes for the dehydrogenases enzymes. The products are 8 acetyl-CoAs, let's say NP plus 7HB, NADH plus 7H plus 7FADH2. And to calculate the energy yield of complete oxidation of palmitate, 1 mole of palmitate, not to forget we have consumed 2 high energy phosphate equivalent to breaking 2 ATPs into 2 ADPs in the activation process. So this is a minus 2. The 7FADH2 will proceed to the electron transport chain. If a malate shuttle is used, this will be equivalent each for 1.5 ATP, so it will be 7 pound 1.5. It will give us 10.5 ATP. The 7 NADH plus H plus using the malate shuttle proceed to the electron transport chain. Each will be equivalent to 2 ATPs, so 7 multiplied by 2.5 is 17.5. The 8 acetyl-CoAs will go to the uric acid cycle, each yielding 10 ATPs per cycle with a total of 80 ATPs. So the total will be 106 ATPs, the full oxidation of 1 molecule of palmitate. Please note that oxidation of fatty acids with even number produces, of course, pure acetyl-CoA molecules as products, while oxidation of fatty acids with odd number will produce also acetyl-CoA in addition to 1 molecule of propionyl-CoA. So the last one will be a 3-atom molecule, the compound that is propionyl-CoA. This propionyl-CoA can be converted, as we know, to succinyl-CoA and can cycle, and then it can gain entry into gluconeogenesis. Propionyl-CoA or propionate is one of the gluconogenic intermediates. Propionyl-CoA is thus considered the only glucogenic part of the fatty acid. How beta oxidation is regulated at the gateway into the mitochondria prior to beta oxidation, where we have carnitine palmitate transferase 1. Its activity is low in the fed state and high in starvation, which makes sense. In starvation, you need energy. You see, as there is no feeding and the glycogen stores are already consumed. So we are going to shift to fatty acids and lipids as a sustained source of energy, and we are going to get that by oxidizing the fatty acids. So the fatty acid regulation will be upregulated, will be activated in the fasting state. While in the fed state, there is enough glucose and glycogen stores are replenished. There is no need for beta oxidation to proceed at a high level. Malonyl-CoA is a protein inhibitor of carnitine palmitoyl transferase 1. And as you remember, malonyl-CoA is an initial intermediate in fatty acid synthesis. So there is a sort of reciprocal control here of both fatty acid biosynthesis, in which malonyl-CoA is an initial intermediate. When fatty acid synthesis is high, there is high concentration of malonyl-CoA. This will inhibit fatty acid oxidation. So in situations where we have active fatty acid synthesis, at the same time fatty acid oxidation will be inhibited through the inhibition of carnitine palmitoyl transferase 1 by malonyl-CoA. In starvation, there is a lot of free fatty acids in the circulation. These free fatty acids act as an allosteric inhibitor of acetyl-CoA carboxylase, so malonyl-CoA will not be formed. And there is also a decreased ratio of insulin to glucagon. Insulin will be low, glucagon will be high. Thus will be this ratio will favor phosphorylation and inactivation of acetyl-CoA. So acetyl-CoA carboxylase is inhibited allosterically, while it is allosteric inhibitor of free fatty acid, and also as a result of phosphorylation, covalent modification, it is inhibited. The net effect is inhibition of acetyl-CoA carboxylase, less product, that is malonyl-CoA, and no inhibition of carnitine palmitoyl transferase 1. That is why in starvation, fatty acid oxidation will be active. On the other hand, in the fed state, there is low levels of circulating free fatty acid, as this free fatty acid will most likely go into esterification. The less inhibition of acetyl-CoA carboxylase in the fed state, insulin concentration is high. Insulin activates and induces acetyl-CoA carboxylate. Activates it through a phosphatase, that is covalent modification, and induces the synthesis of the enzyme itself at the genetic level. At the same time, insulin inhibits lipolysis, that is release of triglycerides from the stores. The net effect in the fed state is that there is high level of malonyl-CoA, which will inhibit carnitine palmitoid transferase 1. So in the fed state, fatty acid oxidation is inhibited. This is glucose here in the fed state. It goes through glycolysis and finally to pyruvate, this formation of acetyl-CoA that can be used because any surplus glucose will be used for fatty acid synthesis and production, and this acetyl-CoA in the first step will be converted to malonyl-CoA by acetyl-CoA carboxylase to build up fatty acids, for example, as we discussed palmitoid in the previous lecture. Acetyl-CoA carboxylase here is activated, as we said, by insulin and inhibited by glucagon, and inhibited also by free fatty acids in the circulation. On the other hand, when there is starvation, free fatty acids will be high, and instead of being esterified, they will inhibit the process. They inhibit the enzyme acetyl-CoA carboxylase, they inhibit lipogenesis in general, and they will favor fatty acid oxidation. How the body oxidizes very long chain fatty acids that are 20 carbon atoms or 22 carbon atom in length. This occurs in the peroxisomes by a modified form of beta oxidation. Acetyl-CoA is also the product. However, there is also production of hydrogen peroxide. Thus, dehydrogenation in the peroxisome is not linked to the respiratory chain. The reducing equivalents here form hydrogen peroxide instead of forming NADH and FADH. There is no production of these coenzymes following oxidation of very long chain fatty acids in the peroxisomes. It ends at the level of an 8 carbon atom, a side residue called octanoyl-CoA, which will go to the mitochondria for further oxidation. The enzymes of oxidation of very long chain fatty acids are induced by high fat diets in some species and the drug chlorfibrate, which is a hypolipidemic drug. We will discuss chlorfibrate and its mechanisms of action shortly. The biomedical relevance of fatty acid oxidation is that it is increased in starvation states and in diabetes mellitus, and it can be associated by ketoacidosis in some instances, but not all the time. Any impairment of fatty acid oxidation will lead to hypoglycemia. Why? Because gluconeogenesis is dependent upon it. When there is a starvation, there is no dietary intake of carbohydrate. Glycogen stores will be consumed. The body will look for an alternative source of glucose that will be synthesizing glucose from non-carbohydrate intermediates through gluconeogenesis. And to operate gluconeogenesis, you need energy. The energy for gluconeogenesis comes from fatty acid oxidation. So if there is an impairment of fatty acid oxidation, there will be no energy for gluconeogenesis, no production of glucose, and it will manifest as hypoglycemia, low levels of glucose in the blood. Carnitine deficiency is encountered in preterm infants in which there is reduced biosynthesis of carnitine or there is renal loss of carnitine. Hemodialysis also will lead to loss and deficiency of carnitine. Deficiency of oxidation, beta oxidation enzymes will manifest as non-ketotic hypoglycemia. Hypoglycemia is not associated with ketone body production. It can also lead to coma and fatty liver, accumulation of fats in the liver. Hypoglycemia, a poison, is found in the unripe fruit of the tree, with manifest clinically as what is called Jamaican food sickness, in which there is inactivation of short and medium chain acyl CoA dehydrogenases. So they will accumulate and lead to these clinical manifestations. Dicarboxylic acid urea, from the name, it is detection of dicarboxylic acid in urine or excretion of dicarboxylic acid in urine. This will be due to lack of mitochondrial medium chain acyl CoA dehydrogenase. Repsum disease is a rare neurologic disorder that is caused by inherited defect of alpha oxidation. It results from accumulation of methylenic acid that comes from phytol, that comes from chlorophyll. So, following ingestion of chlorophyll, this chlorophyll will produce phytol, phytol will produce phytanic acid. Phytanic acid will have to be oxidized by alpha oxidation. If there is no enzymes of alpha oxidation, then this acid will accumulate. Phytanic acid has a methyl carbon on the third carbon, and that is why it is not accessible for beta oxidation. It needs alpha oxidation. Zellweger syndrome is an inherited absence of peroxisome in all tissues, leading to accumulation of very long-chain fatty acids and deficiency of plasma nodules and dysfunction at multiple organ systems. Clofepride is a derivative of fibric acid. It is used in the treatment of some types of hyperlipidemias, particularly hypertriglyceridemia, that is, high level of triglycerides in the blood. It acts via several mechanisms. The overall effect of clofepride is significant reduction in triglyceride levels. That is why it is used to treat hypertriglyceridemia. It has moderate reduction in LDL cholesterol level, potential increase in the good cholesterol, the HDL, and improved lipid metabolism and reduction in the risk of atherosclerosis. The mechanism of clofepride, it increases the activity of extrahepatic lipoprotein lipase. It inhibits synthesis and increases clearance of apolipoprotein B. It is an agonist of peroxisome proliferator activated receptor alpha, PPAR alpha, which leads to the modification of gene expression resulting in increased beta oxidation. We are going to see how these actually work. Increasing the level and activity of extrahepatic lipoprotein lipase will promote hydrolysis of triglycerides that are present in the very low density lipoproteins and chylomicrons, which will lead to reduction in triglycerides in blood. Decreasing synthesis and increasing clearance of ApoB. Apolipoprotein B is structurally part of VLDL and LDL. So they will be reduced, and this will eventually lead to reduction of their level and will manifest overall as less risk of atherosclerosis. PPAR alpha activation will activate increase transcription of genes, particularly of the enzymes of beta oxidation, such as carnitine palmitoyl transferase 1. This will lead to increased beta oxidation in the peroxisome mitochondria and less accumulation of triglycerides in the liver and circulation. And before we end, there is a question for you. Is lipolysis synonymous with fatty acid oxidation? Yes or no? Justify your answer. Thank you very much. See you next time.